My name is Jason Aliotta, and I'm the Associate Program Director of the Pulmonary Critical Care Fellowship. I became interested in looking at the ability of hematopoietic stem cells to differentiate into pulmonary epithelial cells. How is it that these stem cells all of a sudden were able to turn into lung cells? And what we found was that lung cells after injury were releasing small particles called extracellular vesicles. And these vesicles contained proteins, RNA, DNA, a variety of different things, microRNAs as well. And what we found was that stem cells internalized these extracellular vesicles, and after internalization, started to produce or started to express pulmonary epithelial cell specific genes and proteins as well. So we found this to be sort of an interesting interaction between an injured and potentially dying cell with a cell that had the ability to regenerate the pulmonary epithelium. I was interested in looking at the role of these extracellular vesicles and what role they played in pulmonary vascular disease. We had a mouse injury model. It was a mouse model that mimicked certain features of pulmonary hypertension. And what we found was that if we extracted these vesicles from the blood or from the lungs of mice with pulmonary hypertension and infused them into healthy mice, those mice would then eventually develop signs and symptoms of pulmonary hypertension. And I thought, wow, this is really interesting. How is this happening? Right now, where my research has taken me is that I'm in the process of applying for R01s, and the focus of my R01s is to look at the interactions of extracellular vesicles that are shed from the injured pulmonary vascular endothelium in the setting of pulmonary hypertension, finding out what these extracellular vesicles are specifically doing. Are they going back to the pulmonary vasculature and creating pulmonary vascular or inducing pulmonary vascular remodeling directly? Or the alternative hypothesis that we're working on and looking at right now is that these vesicles are actually homing to the bone marrow and inducing bone marrow progenitor cells to differentiate into endothelial progenitor cells, which are then homing to the lung and inducing pulmonary vascular remodeling. And this is really the focus of the grants that I've applied for at this point.